There is a piece of linen cloth kept inside a climate-controlled silver casket in the Cathedral of St. John the Baptist in Turin, Italy. To look at it, you would not immediately understand why it has captivated scientists, historians, theologians, skeptics, and believers for more than 600 years. It is 14 feet long and 3.5 feet wide. It is ivory-colored, ancient, faintly discolored in places, and on its surface, visible to the naked eye as a pale brownish-yellow impression that grows stranger and more inexplicable the closer you look, is the image of a man who has been beaten, a man who has been scourged across his back and legs more than 100 times, a man whose head bears wounds consistent with a ring of piercing thorns, a man with nail wounds not through his palms, the way medieval artists always painted crucifixion, but through his wrists, the way Roman executioners actually did it, a man with a massive wound in his side, a man who appears to have been wrapped in this cloth in death and then left an image inside it that no one, not a single scientist, not a single forger, not a single laboratory with the most advanced technology on Earth, has ever been able to explain, replicate, or convincingly account for. This is the Shroud of Turin. And in the first months of 2026, after decades of accumulating scientific investigation, it gave up something that stopped researchers cold. It gave up its DNA. And what that DNA revealed about where this cloth has been, who touched it, what it carries, and what it may have once wrapped, left the scientists who read the results staring at each other across a laboratory in northern Italy with nothing adequate to say. In 1988, the Catholic Archbishop of Turin authorized the removal of a small sample from a corner of the shroud. 3 of the world's most prestigious radiocarbon dating laboratories received portions of that sample: the University of Arizona, Oxford University, and the Swiss Federal Institute of Technology in Zurich. All 3 were asked to perform carbon-14 dating, the standard method for determining the age of organic material. All 3 came back with results that fell between 1260 and 1390 AD, medieval. The cloth, the laboratories concluded, was a medieval forgery. It was not 2,000 years old. It was not the burial shroud of Jesus Christ. It was a piece of linen made sometime in the late Middle Ages in Europe. For many people, that ended the conversation. The science had spoken. Case closed. Except the case refused to stay closed, because carbon dating, like every scientific method, is only as good as its sample. And in the years following 1988, a series of investigations began to raise increasingly serious questions about whether the sample tested in 1988 was actually representative of the original cloth at all. The corner from which that sample was taken was, multiple textile experts pointed out, precisely the part of the shroud that had been most heavily handled over the centuries, most often touched by pilgrims and clergy, most likely to have been repaired with newer thread woven into the existing fabric. Ray Rogers, a chemist who had been part of the original 1978 STIRP investigation, the most comprehensive scientific examination of the shroud ever conducted, performed his own analysis of the 1988 sample area and found chemical signatures consistent with a medieval repair patch rather than the original ancient linen. If he was right, the laboratories had dated the patch, not the cloth. Then in 2022, something happened that changed the scientific debate completely. Dr. Liberato De Caro of Italy's Institute of Crystallography, working at the National Research Council's laboratory in Bari, published the results of a study using an entirely different dating method. This method is called wide-angle X-ray scattering, or WAX-S. Instead of measuring radioactive carbon decay, which is susceptible to contamination, WAX-S measures the degree of natural degradation in the cellulose fibers of linen. Linen is made from flax, and the cellulose chains and flax slowly break down over time in a way that correlates with age. The older the linen, the more degraded those chains. You cannot fake this degradation with a repair patch. You cannot fake it with contamination. The molecules simply change over time, and WAX-S reads that change. De Caro and his team studied 8 tiny samples from the shroud, each no larger than 0.5 millimeter by 1 millimeter. So small they were essentially invisible to the naked eye. They compared the X-ray scattering profiles of those samples against a reference collection of linen fabrics with known, documented ages spanning from 3000 BC to 2000 AD. And then they compared it against something very specific, a piece of linen whose age is known to the year because it comes from an archaeological site whose destruction date is recorded in history. That linen came from the fortress of Masada in Israel, the fortress that fell to the Roman siege in 74 AD, meaning the linen in that fortress was last used approximately 2,000 years ago. The WAX-S profile of the Shroud of Turin matched the Masada linen. Not approximately, not roughly. The experimental results were, in De Caro's own scientific language, fully compatible with the Masada sample. The degree of natural aging in the cellulose of the shroud was consistent with a cloth that is approximately 2,000 years old. The peer-reviewed paper, which received enormous international media coverage when it was highlighted in August 2024, concluded with a statement that struck the scientific community like a thunderclap. The experimental results are compatible with the hypothesis that the Turin Shroud is a 2,000-year-old relic, as supposed by Christian tradition. Not proven, not confirmed, but compatible, meaning that when you remove the contaminated corner and use a method that cannot be fooled by patching, the cloth behaves exactly the way a 2,000-year-old piece of linen should behave. This is the context into which the 2026 DNA findings arrived. Not a settled question, not a closed debate, a debate that had been ripped wide open again by a conflict between two scientific methods pointing inexactly. The DNA investigation of the Shroud actually began earlier than most people realize. The original samples that made the 2026 study possible were collected not recently, but in 1978, during a period when the Shroud was examined by a team of American and international scientists known as STURP, the Shroud of Turin Research Project. STURP was the most intensive scientific examination the cloth had ever received. 33 scientists from various disciplines spent 120 continuous hours examining the Shroud with an array of instruments. Among the researchers who were part of that period was Professor Pier Luigi Baima-Bologna, a forensic medicine specialist from Turin. In the 1980s, Baima-Bologna became the first scientist to formally identify the substance on the Shroud's surface as genuine human blood. Specifically, human blood of the AB blood group. He preserved samples from his work. Those samples, held in careful storage for more than 40 years, became the raw material for the 2026 study. Baima-Bologna died in November 2025, before the paper he co-authored was published. His life's work, preserved in those samples, spoke after him. The 2026 study was led by Dr. Gianni Barcaccia, professor of genetics and genomics at the University of Padua, along with an international team of researchers. Barcaccia had been working with Shroud DNA since 2015 when he published a major study in Nature Scientific Reports. The first systematic DNA analysis of the cloth. That 2015 study was already remarkable. It identified plant species and human mitochondrial DNA haplogroups on the Shroud's surface, giving the first genetic hint of where the cloth had been and who had touched it. But the 2026 study, using substantially more advanced sequencing techniques on material from the 1978 collection, went far deeper. And what it revealed was extraordinary on every level. The most fundamental finding of the 2026 analysis concerns the geographic origin of the human DNA on the Shroud. When Barcaccia's team ran their sequencing and mapped the mitochondrial haplogroups they found against the global population database, they discovered that the human genetic material on the Shroud does not look like it came from medieval France or medieval Europe. It looks like it came from somewhere else entirely. The dominant genetic signature, more than half of the human DNA on the cloth, clusters with populations from the Near Ear, Saint, and Middle East. Specifically, the researchers confirmed the presence of haplogroup H33, which the study describes as prevalent in the Near East and common among the Druze population. The study notes precisely why this matters. The Druze population shares common genetic ancestry with Jews and Cypriots and has historically intermixed with other Levantine populations, including Palestinians and Syrians. The Middle Eastern genetic signature on the Shroud is not a minor trace. It is the dominant profile. And it is the profile that you would expect if a cloth had spent significant time in the region that is today Israel, Jordan, Lebanon, and Syria, the precise geographic area that would align with the Shroud's claimed history as a burial cloth from 1st century Jerusalem. Europeans, by contrast, represented less than 5.6% of the human DNA on the cloth, according to Barcaccia's analysis. For a cloth that has been kept in Europe for the past 700 years, handled by European clergy, venerated by European pilgrims, restored by European craftswomen, and stored in European cathedrals. The fact that European DNA is nearly absent while Near Eastern DNA dominates is one of the most scientifically arresting results the study produced. It implies that the cloth's fundamental biological imprint was established before it arrived in Europe, and that the DNA absorbed during its European centuries barely registers against a pre-European genetic baseline. But the Near Eastern DNA finding, astonishing as it is, was not the result that left researchers speechless. That result came from a different part of the DNA map, because the 2026 study also confirmed and expanded upon a finding that had first appeared in Barcaccia's 2015 analysis, a finding that no one had adequately explained in the 11 years since it first appeared, and that became dramatically more significant in 2026 with better sequencing and a larger data set. Approximately 38 to 40% of the human DNA on the Shroud belongs to mitochondrial haplogroups that are characteristic of populations from the Indian subcontinent. Haplogroups designated M39, M56, R7, and R8, Genetic lineages that are concentrated in India, Pakistan, and surrounding South Asian populations appear on the shroud at levels that cannot be explained by random contact or modern contamination. Think carefully about what that means for a moment. A cloth that, if authentic, was used in 1st century Jerusalem, that first appears in documented European history in France in 1354, and that has been stored in European cathedrals ever since, carries a genetic signature that is nearly 40% South Asian, and only 5% European. In the words of the research team, these results raise the possibility of an Indian manufacture of the linen cloth. The linen itself, the raw material from which the shroud was woven, may have originated in India, not in the Middle East or Europe. And the people whose DNA most permeates its fibers are not the European pilgrims who kissed and touched it in medieval France and Renaissance-era Italy. They are people whose genetic lineage traces back to the subcontinent. This finding sent scientists scrambling for historical context, because it is not as implausible as it might initially seem. India in the ancient world was not isolated from the Mediterranean. The Roman Empire maintained active and extensive trade relationships with Indian merchants through networks like the Silk Road and the Indian Ocean trade routes. Indian textiles, particularly high-quality linens and cottons, were known to be exported to the Roman world. Archaeological evidence of Indian origin goods has been found at Roman sites across the Mediterranean. A burial cloth woven in India and used in 1st century Roman-controlled Judea is not historically impossible. It is, in fact, consistent with everything we know about the trade networks of that era. But the DNA on the shroud did not stop at human origins. The 2026 study also conducted what is called metagenomic sequencing, reading not just the human DNA, but the entire biological record of everything on the cloth: plants, animals, microorganisms. The result was one of the most complex biological portraits ever assembled from a single ancient artifact, and each layer of it contributed something new to the story. The plant DNA analysis identified at least 19 different plant species, whose genetic material is present on the shroud. Several of these are species native to the Mediterranean basin, which you would expect given the cloth's known European history. But others are species with primary origins in Asia, in the Middle East, and here is where the results became particularly startling: in the Americas. Plant species that originated in the Americas and were not introduced to the Old World until after 1492 are genetically present on the shroud's surface. Corn, banana, peanut, and other New World species appear in the botanical DNA. Botanists on the team were careful to note that this is almost certainly explained by post-Columbian contact. Pilgrims or handlers bringing these materials to the cloth after the Columbian exchange began in the late 15th century. But it illustrates something important. The cloth is genuinely a living record of every environment it has passed through, every hand that touched it, every breath of air it absorbed across what may be 2,000 years of existence. The animal DNA told a parallel story. Researchers identified genetic material from cattle, pigs, chickens, sheep, goats, horses, deer, rabbits, cats, and dogs. From fish species, including gray mullet, Atlantic cod, and ray-finned fish, from marine crustaceans, from flies, aphids, and arachnids. Every animal whose presence near this cloth, in a market, in a stable, in a church storage room, in a medieval city street, left a trace of shed fur, a flake of scale, a fragment of wing, is now written into the genetic archive of the shroud. The cloth has been soaking up the biological world around it for as long as it has existed, and modern DNA sequencing can now read that record in detail that would have seemed miraculous to scientists even 20 years ago. But perhaps the most scientifically significant non-human DNA finding from the 2026 study involves microorganisms, specifically the microbiome that has colonized the shroud's surface over its lifetime. The researchers identified bacteria commonly found on human skin, including Cutibacterium and Staphylococcus, which are transferred by touch and reinforce the picture of extensive human handling. They found fungi, including molds, yeasts, and other organisms, consistent with the cloth being stored in varying conditions of temperature and humidity over centuries. And they found something else, something that, when it appeared in the sequencing data, made researchers look twice at what they were reading. They found communities of archaea, a domain of single-celled microorganisms distinct from bacteria, specifically adapted to extremely saline environments. Salt-tolerant microorganisms, the kind of microorganisms that thrive in places like the Dead Sea. The Dead Sea sits in the Jordan Valley, less than 20 miles from Jerusalem. Its salt concentration is approximately 10 times that of normal seawater. Nothing resembling Dead Sea archaea should be present on a cloth that has spent its entire existence in Europe. Their presence is a biological fingerprint, an ecological stamp left by an environment that is specific, identifiable, and geographically precise. It suggests, as the Vatican News outlet reported when the study became available in April 2026, that the shroud passed through the Middle East, and spent time in proximity to an environment with the chemical signature of the Dead Sea region. The microbiome of the cloth carries the memory of a place that is 14 inches above sea level on the eastern edge of what is now Israel. These DNA findings, Near Eastern dominant haplogroups, South Asian linen origins, Dead Sea microbiome, do not stand alone in the scientific record on the Shroud. They land into a forensic landscape that had already been accumulating evidence pointing in the same direction, piece by piece, from multiple independent lines of investigation. And taken together, the picture those lines build is one of the most remarkable convergences in the history of physical science. Start with the blood. Baima Bologne's identification of blood group AB on the Shroud in the 1980s was confirmed by subsequent analysis. Blood type AB is rare. It is found in only 3 to 5% of the world's population. It is disproportionately common, however, in Jewish and Middle Eastern populations, where its frequency can be 2 to 3 times higher than in Western European populations. The blood on the Shroud is not just type AB. Forensic analysis of the bloodstains, particularly the work done during the Esturpi investigation and in subsequent peer-reviewed studies, found elevated levels of bilirubin, a compound produced in large quantities by the body under conditions of extreme physical trauma. Bilirubin is what gives bruises their yellow color. It accumulates rapidly in the blood of a person being beaten, flogged, and tortured over a period of hours. The bilirubin level in the Shroud's bloodstains is consistent with a person who experienced exactly that kind of prolonged, severe physical abuse before death. It is not consistent with a person who died peacefully or quickly. It is consistent with crucifixion. The wounds recorded in the Shroud's image have been analyzed by forensic pathologists and trauma surgeons who have spent careers studying what the human body looks like after extreme violence, and what they find in the Shroud consistently stops them. There are over 100 individual wound marks across the back and the legs, arranged in a pattern consistent with a Roman flagrum. A whip with multiple leather thongs, each tipped with small lead weights shaped like dumbbells. The pattern of the marks suggests the blows came from 2 different angles, consistent with 2 soldiers of different heights striking alternately, which matches what historians know about Roman scourging practice. The nail wounds are in the wrists, not the palms. Medieval artists, without exception, painted crucifixion wounds through the palms because that is where they assumed the nails went. But a forensic engineer who analyzed the mechanics of crucifixion showed that nails through the palms cannot support the weight of a human body. The hands would tear. The nails had to go through the wrists, through the space called the space of Destot, to provide structural support. A medieval forger, working from the artistic conventions of his time, would have put the wounds in the palms. The Shroud's wounds are in the wrists, the anatomically correct location that a medieval forger would not have known. There's the question of pollen. A Swiss criminologist and botanist named Max Frey spent years collecting tape samples from the Shroud's surface and identifying pollen grains. He found pollen from species native to the region around Jerusalem, to Anatolia, and to Constantinople. The precise sequence of locations that the Shroud would have passed through if it traveled from Jerusalem through what is now Turkey to France, following the historical routes available in the early medieval period. Some of the pollen species he identified are associated with springtime flowering in the Jerusalem area, which is when Passover and the death described in the Gospels occurs. Later, botanist Avinoam Danon of the Hebrew University of Jerusalem independently examined the Shroud and identified plant images, not just pollen, but faint impressions of entire flowers, pressed into the surface of the cloth. One of those flower images, belonging to a species called Zygophyllum domosum, grows only in a very specific geographic range that includes the area around Jerusalem, the Jordan Valley, and the Sinai. It does not grow in medieval France. It does not grow anywhere in Europe. And there is the Sudarium of Oviedo. This is a smaller cloth, measuring about 83 by 53 centimeters, that has been continuously documented since the 7th century in the Cathedral of Oviedo in Spain. Historical records trace it back to 1st century Jerusalem. It is believed to be the cloth described in the Gospel of John that was used to cover the face of Jesus immediately after his death, before the body was wrapped in the larger burial cloth. The Sudarium has been in a known documented location since 616 AD. Nobody disputes its historical chain of custody, and when forensic scientists compared the Sudarium to the Shroud, they found more than 120 matching points of correspondence in the bloodstain patterns, the exact same AB blood type, the same pattern of blood flow consistent with a specific head position, the same arrangement of wounds around the head. 2 cloths from 2 completely independent historical chains of custody, telling an identical forensic story about the same body. The image itself is perhaps the deepest mystery of all, and it connects to the DNA story in a way that is not immediately obvious. The body image on the Shroud, that pale, ghostly impression of a crucified man, is not a painting. This was established definitively by the Stirp investigation in 1978. There is no pigment, no dye, no chemical on the image areas. The color comes from a very thin layer of oxidized, dehydrated cellulose on the very topmost fibers of the linen threads. A change in the organic structure of the fiber itself, not a substance applied from outside. The image does not penetrate the cloth. It is only on the outermost surface, thinner than a human hair, and it fades with depth at a rate that no known painting or chemical staining technique can replicate. When computer scientist Pete Secondo at the Jet Propulsion Laboratory ran the Shroud's image through a VP8 image analyzer, a device designed to convert light intensity in a photograph into topographic elevation data, the image encoded three-dimensional spatial information. Flat photographs, paintings, and prints produced distorted noise when run through this analyzer. The Shroud produced a perfect, undistorted three-dimensional relief of a human body. The image contains spatial information that a two-dimensional representation cannot possess. No one has yet explained how it got there. Scientists who have attempted to replicate the image using medieval techniques have all failed. Scientists who have attempted to reproduce it using modern radiation techniques have produced partial approximations, but never the combination of characteristics: surface-only deposition, no pigment, three-dimensional encoding, and uniform distribution across a full body front and back that the Shroud exhibits. A team of Italian scientists from ENEA, the Italian National Agency for New Technologies, Energy and Sustainable Economic Development, concluded in 2011 that the image formation on the Shroud required an intensity of vacuum ultraviolet radiation that did not exist in the medieval world and cannot currently be produced by any device except a free electron laser. They were not claiming a miracle. They were saying, in the careful language of scientists, that they could not account for the image by any mechanism they could identify. It is into this accumulation of unexplained evidence, the whack-kisses dating, the blood forensics, the pollen, the wound analysis, the sudarium correspondence, the unexplainable image, that the 2026 DNA results landed, and the scientists who read them understood immediately that the DNA does not prove the Shroud is the burial cloth of Jesus. No DNA test could prove that. There's no DNA sample from Jesus to compare against. There's no reference genome. Even if the dominant human haplogroup on the cloth matches a first-century Jewish man from the Jerusalem area, that description fits millions of people who lived in that time and place. The DNA tells you about origins, about journeys, about who touched this cloth. It cannot tell you whose blood soaked into it 2,000 years ago. What the DNA does do, and what is quietly extraordinary when you consider the totality of what has been found, is add one more independent line of evidence pointing in the same direction as every other line of evidence that has been seriously investigated over the past 5 decades. The dominant human genetic signature matches the Near East, the microbiome matches the Dead Sea region, the pollen matches Jerusalem in spring, the blood type and bilirubin match severe trauma. The wound patterns match Roman crucifixion. The age estimate from Waxess matches 1st century linen. The botanical evidence matches 1st century Israel. The sudarium correspondence matches a specific body. The image formation defies current scientific explanation. None of these things individually proves anything. Scientists are careful about this. Dr. Barkacci himself, presenting the 2026 findings, was explicit. The DNA mixture is too contaminated and too complex to connect the material to one original individual. The evidence shows contact, it shows history, it shows journey, but not identity. Even among researchers who find the cumulative case compelling, there is careful insistence that no single test and no single finding should be read as proof of anything beyond what it directly measures. And yet there is a question that hangs over the laboratory results, over the X-ray scans, over the forensic reports, over the pollen studies, over the microbiome data, that science in its current form cannot answer and perhaps will never be able to answer. Because the Shroud of Turin, as described by the Harvard study that called it the most studied artifact in human history, has now been subjected to more scientific investigation than any object in existence. Billions of dollars of technology, hundreds of the world's finest scientific minds, decades of research. And the answers keep coming back the same way. Old, Middle Eastern, covered in the blood of a tortured man, stamped with an image no one can replicate, carrying the genetic echoes of South Asia, the Near East, the Dead Sea, and nearly 2,000 years of human hands. The scientists stared at the 2026 results and found themselves in the position that science least enjoys occupying, in the presence of evidence they cannot fully explain, pointing toward a conclusion they cannot scientifically confirm. What they could say, and what they did say, is that the DNA record of the Shroud of Turin is, in the precise scientific language of Dr. Barkacci's team, a diverse mosaic of genetic traces. a biological autobiography of a cloth that has been somewhere and touched by someone and kept in an environment that has left its chemical signature in the microorganisms still alive in its fibers. A cloth that has carried its secrets in its molecules for 2 millennia, waiting for a science advanced enough to read them. And now that science has read them, and the reading left them speechless.